Hello students, I am Dr. Mandeep Kaur from Guru Nanak College, Sri Muktsar Sahib, and today we will talk about photosynthesis. Now this is a reaction you have studied in lower classes as well. 6 carbon dioxide plus 6 water in the presence of sunlight, it gives glucose plus 6 oxygen. However, this is not such a simple reaction. It is quite a complex thing, and we will talk about it in detail how it happens. Now you must all be intrigued by how does these simple looking leaves, which are green in color and in the presence of sunlight, how do they make food which is on which all the organisms, plants as well as animals, they are dependent. Even some fungi are dependent on them. So how does it occur? We all know that the plants, they take carbon dioxide through their stomata. Carbon dioxide is released by the animals as well as plants during respiration. It is present in the atmosphere also. Then they take up water through their xylem and roots, etc. Then sunlight is available, and for which we take for granted always. And then they form the sugars which are taken up by the used up by the plant as well, and then taken up by other animals, other plants, and then they also release oxygen, right? So how does all this happen together? So photosynthesis basically occurs in the chloroplast. This is the structure of a chloroplast. You all know it is a double membrane structure, semi-autonomous structure. It has thylakoids which are stacked together in grana, and then it has rest of the place is called stroma. So these thylakoid and the stroma words we will be using again and again. So what happens? The different reactions, different enzymes, all of them are compartmentalized in different parts of the chloroplast. So thylakoids will have the reactions of the light reaction in them, and the dark reactions, they occur in the stroma, right? However, the terms light reaction and dark reaction, these are misnomers. We should call them light dependent reactions, and dark reactions should be called light independent reactions, right? So what happens is in the presence of sunlight, the chlorophyll which is present inside the thylakoids, this is stimulated and excited, and then water is converted and hydrolyzed into oxygen. Then the excited electrons, they go through various reactions to ultimately form ATP and NADPH. So the main purpose of the light reactions is to form ATP and NADPH, and in the process water is converted into oxygen. So oxygen is just a byproduct which eventually helped in making the earth as an oxygen containing environment. We all know that earlier the atmosphere in the earth, it was not having oxygen, right? That is why we had anaerobic bacteria, etc. So this oxygen evolving concept was started in some cyanobacteria, and then this oxygen started being used up, and slowly the further mechanisms happened, and the evolution happened, right? So the main function of the light reactions is to form ATP and NADPH. These ATP as well as the reducing power of NADPH, this is used up in the next light independent reactions, which are we call Calvin cycle, where RUBP, that is the ribisco 1,5 bisphosphate in the presence of carbon dioxide is now utilized, and the enzyme ribisco, that is ribisco is ribulose 1,5 bisphosphate carboxylase oxygenase. So this converts it into 3-PGA, which is converted into glyceraldehyde 3-phosphate, which converts it into sugar, and the rest of the cycle is for regeneration of RUPP. So this reaction, that is the light independent reactions of Calvin cycle, they occur inside the stroma. So now how all these things occur, we will do in detail. First of all, we should know about the photosystems. There are two photosystems in the chloroplast, that is photosystem 1 and photosystem 2. Photosystem 1 is called photosystem 1 because it was discovered earlier, and later on the one which was discovered, it is called photosystem 2. Now, photosystem 1 has major chlorophyll molecules, then accessory pigments also like carotenoids, etc. And their absorption spectrum is at 700 nanometers. So that is why it is also called P700, right? And PS2 or the photosystem 2 has its absorption spectrum at 680 nanometers. So it is also called P680, right? Then what happens is when the sunlight is falling on the leaves and chlorophyll are there inside the chloroplast, and this all is occurring in the thylakoid membrane. So these are the various chlorophyll molecules, and all of them, they get excited, and they get excited. The electron is transferred to the next chlorophyll until it reaches the main trap center. These are all the, this whole thing is called the trap center, right? And then what happens is uh, excites, the excited electron is taken up by the pheophytin. Pheophytin gives its electron to plastoquinones QA and QB. These go through Q and QH2 cycle. Then it goes, the electron is taken it by cytochrome B, iron sulfur protein, and cytochrome F. So this is the cytochrome complex, which is then taken up by plastocyanin. Plastocyanin gives its electron to P700. This is also excited and taken light, excited by the sunlight. Then P700 gets excited and its electron is given to, taken up by the iron sulfur proteins, followed by ferredoxin, and the final electron acceptor in this system is NADP positive, and then it forms the NADPH. So this is about the Z scheme. So it looks like a Z, so it is called Z scheme of photosystems, right? So we will have another depiction. So this is P680. First of all, P680 is initiated, and then it gives its electron to the various electron transport system we have already done over here. That is quinones, then plastoquinone, then cytochrome B6F, and then finally plastocyanin. Then P700 will be excited, and it gives its electron further through uh, ferredoxin, etc. Finally to NADP positive, which forms NADPH. Now this NADPH will be um, entering the stroma, and it will take part in the Calvin cycle. Now when this P680 is excited by the sunlight, there is another thing which is happening, that is the water hydrolysis. During this water hydrolysis, two electrons are released and one half oxygen and two protons. So these protons will also function. We will just now see. So this is how what do you call it, oxygen is released. So first step is basically the release of the oxygen, right? And now this is the thylakoid membrane, and inside is the thylakoid lumen, and outside is the chloroplast stroma, right? So here again the thing is the same, the depiction is different, but we get to see a new thing over here. So oxygen evolution complex is there, water is given, water is hydrolyzed into oxygen and protons. These protons will move, and there will be a positive charge inside the thylakoid membrane. So PS2, then plastoquinone, again protons will move in this point also. And then what happens is cytochrome B6F, then plastocyanin. You should also remember the place where these different things are present, like plastocyanin is present inside the thylakoid lumen. This PS2 and cytochrome BF and PS1, these are across the thylakoid membrane, right? However, this ferredoxin and FNR, which is ferredoxin NADP reductase protein, these are present in the stroma, and then that is why NADPH is released inside the stroma. This is required in stroma and it is already being formed inside the stroma only, right? And then due to all these proton movements and increase of proton inside the thylakoid lumen, you have proton motive force. Due to this proton motive force, what happens is the proton functions as a rotor and then they will move the proteins of the ATP synthase, and these proteins will move and eventually ADP plus phosphate will form ATP. So the two main things which we required, that is NADPH and ATP, for the next step, that is Kelvin cycle, they are being formed over here like this, right? And oxygen is just a byproduct. So inside the thylakoid lumen, this is another depiction so that you can understand how all this is happening. So this is one thylakoid and this is the lumen, and outside we have the stroma of the chloroplast. So this is PS1, then hydrolysis of water is occurring, electrons are moving through plastoquinone, then cytochrome B6F, then plastocyanin, which is located inside the thylakoid lumen, then PS1, then ferredoxin and FMM and NADPH is formed, right? And protons, you can see during the electron transport chain, protons are gathering inside the thylakoid lumen. You get a proton motive force, ATP synthase is activated and you get ATP. So NADPH as well as ATP have been formed. Next comes the Kelvin cycle. So Kelvin cycle we will do in the next lecture in detail. Here we will just take an overview of this. So during this is the whole chloroplast. These are the thylakoid and grana. So from water we have got oxygen, light dependent reactions have occurred, and you have got the NADPH as well as the ATP. These will be now utilized during the Kelvin cycle in the presence of Rubisco enzyme as well as carbon dioxide which has been taken in from the stomata, and sugar will be released and utilized for various purposes. I hope you have understood the first part, that is the light dependent reactions. Light independent reactions will be done in the next lecture. Thank you.